Thank you, Ryan. Good morning, everyone. For those of you that don't know me, I'm Matt Daugaard. I'm part of the research faculty at the Vancouver Prostate Center. I'm also the faculty head of the molecular pathology and imaging core facility there. So we are handling many of this, tissue samples and liquid biopsies that come over from here. And then we perform research tasks and research analysis on these samples for all our research personnel. In my academic research lab, we are primarily working on two different aspects of cancer research. One half of the lab is working on DNA damage and repair pathways and drug development targeting those pathways. And the other part of the lab is working on sugar, glycosaminoglycans in cancer. Also in the context of identifying vulnerabilities in cancer that we can target with different types of therapeutic approaches. So I have a few disclosures here. Over the years, I've co-founded a number of companies based on the research that we have done. For this particular talk, it will be the first two companies up there, VAR2 Pharmaceuticals and Prostate Diagnostics, that will be relevant in this context because those are the companies that are currently moving forward the technology that I'm talking about today. All right, I put down some learning objectives. So what I really hope to do today is to give you a good understanding of glycosaminoglycans in cancer, why they're important, how they are regulated, deregulated, and then also give you an idea about some of the targeting strategies that we are applying to targeting glycans in cancer. They're a bit different in some ways from traditional targeting approaches. And then I will also try to go over all the roadblocks that we have had over the years in developing drugs for glycans. Some of them are research-based roadblocks, but many of them are also business-wise roadblocks and roadblocks put in by pharmaceutical partners. So, okay. So, glycosylation is a secondary modification to proteins, but it can also be on lipids, other biomolecules. All life can glycosylate, at least all eukaryotic life. So from yeast to humans, plants, they can all glycosylate. And glycosylation is a secondary modification that changes the properties of the proteins. So you can think of it as a phosphorylation or a ubiquitination or an acetylation. All the different ways a protein can be modified to change function. Glycosylation have similar roles, but they also have structural roles. They impact stability, and they also create what we call the glycocalyx that surrounds all living cells in our body and in all organisms. It's a coat of glycosylations, glycosaminoglycans that basically act as a shield around cells and protect cells from pathogens, immune cells, regulate immune cells, have many roles in a normal physiology. The glycosaminoglycans are always linear structures. So it's sugar rings that are attached after each other. And if you look at this here in the middle, proteoglycans, those are the proteins that are modified with glycosaminoglycans. And those proteoglycans, we know of approximately 30 to 35 bona fide proteoglycans in cells that can be modified with a variety of different types of glycosylation, such as heparan sulfate or chondroitin sulfate, dermatan sulfate, etc. But there are also many proteins that are not normally modified, that becomes modified with glycosylations in diseases, such as cancer. Okay, so glycosylation in cancer is not a new invention. It has been observed over the past, I would say, 4 decades at least, sporadically in most types of cancers. What you see is the glycocalyx that we just looked at before is getting thicker, so you get an increase in thickness of the glycocalyx. And then you change the composition of the glycosaminoglycans to facilitate cancer cell survival. And that is both in the context of proliferation, migration, and motility, but it's also protecting from immune cells. So it's part of why some tumors is immune cold. The immune cells cannot penetrate because of the thick glycocalyx. So the breakthrough we got, and that this is, I think it's 13 years ago now, where we caught a break in how we can look at the cancer glycocalyx. And what we, by coincidence, found out was that the malaria parasite, or a specific subtype of the malaria parasite, express a lectin. A lectin is what we call proteins that have high affinity to glycans. So natural occurring proteins that bind specific glycan motifs. So what happens here is that the malaria parasite, and that type of malaria parasite that causes pregnancy-associated malaria outbreaks in Africa, they express a protein called VAR2-CSA that specifically binds to a chondroitin sulfate glycosaminoglycan that is only present in the placenta in normal adults. And that makes it possible for the parasite to sequester in the placenta during pregnancy, and that is the basis of malaria outbreaks in the placenta. So that protein that binds chondroitin sulfate is called VAR2-CSA. It's a big protein. It's a 350 kilodalton protein coated in the genome of the malaria parasite, but put on the surface of infected red blood cells so they can anchor in the placenta, sequester there. So when we kind of saw this coming up back in many years ago, we reasoned that this type of chondroitin sulfate that you have in the placenta might be the same type of chondroitin sulfate that you find deregulated in cancer. And that is because the placenta and tumors have so many things in common. They are both, organs that have to proliferate rapidly. The placenta has to become a 500-gram organ in 40 weeks. And it has to invade the neighboring tissue. So it has an invasion, proliferation common with tumors. And then it also has to protect itself from the immune system. So immune evasion is also part of it. So for that reason, this is actually the first experiment ever done with this. So we took infected red blood cells from pregnant women in Tanzania and added them on to different types of tumor cells. And all the small dots you see here around, that's the infected, red blood cells. And what we saw, they all bound very, very tightly to tumor cells. That was the first indication that the chondroitin sulfate deregulation you see in cancer might be the same as you actually have in the placenta. And if that's true, then we have a reagent from the malaria parasite that we can use molecules to interrogate that modification in cancer. This was just to show that if we took purified placental chondroitin sulfate and added it to this mixture, then we could completely block the interaction between the infected erythrocytes and the tumor cells, indicating that that was the mechanism at that point. So this malaria protein, we shortened that down to 75 kilodalton because that's the minimal binding region to chondroitin sulfate. And because this type of chondroitin sulfate is now present in placenta and in cancer, we named it Oncofetal chondroitin sulfate, just to distinguish it from normal chondroitin sulfate. This malaria protein binds any type of tumor cells in culture, and you can compete it out with purified chondroitin sulfate. It also binds BEVO, that's a placental cell line, of course, but it doesn't bind normal cell lines, as we would expect from the malaria tropism. So over the years, we have interrogated this modification in most cancer indications. And all solid tumors are positive, or at least a good portion of them. So this is a pan-cancer modification that comes up when tumors develop. And in some tumor indications, it's an early event. In other tumor types, it's a little bit later event. And one thing that we found out here that is very important also for the targeting strategy is that this is a stromal target. It's expressed on the surface of tumor cells, but it's also present in the entire stromal compartment of the tumor, because that's where the proteoglycans, they integrate in their network. Okay. So just this is one of the first really deep characterization studies we did in bladder cancer. And what we could see here is that bladder cancer, first of all, they express the modification in a progression-dependent manner. We also saw that patients that relapse after neoadjuvant chemotherapy, they come back with high expression of this modification. It's not that it's not expressed before, but the tumors that relapse on cisplatin, they have much more of this modification than before. And then the postdoc at the time, Roland Seiler, he developed this model here that you can see up here, where he took a cisplatin naive human bladder cancer line, and then he put it into the bladder wall of the mice using ultrasound. And then he subjected it to cisplatin. And every time the tumor came back on cisplatin, he passaged it into a new mouse. And after 4, 5, 6 generations of this, we were working with a completely cisplatin-resistant tumor in the mice that we could then try to treat with a therapeutic based on the malaria protein. And what we made here was this, a VDC, we called it, a VAR2 drug conjugate. So it's like an antibody drug conjugate, but it's just not an antibody. It's a recombinant part of the malaria. And then we attached drugs to this. At that time, we used a drug from, at that time, Kantara, or Kairos Therapeutics here in Vancouver. They later got bought up by Zymeworks. So now that platform sits with Zymeworks. And that drug here was able to very nicely inhibit tumor growth in these mice, these tumors that were completely cisplatin resistant. And it was not just bladder cancer. So any solid tumor that were positive for oncofetal chondroitin sulfate were sensitive to this type of molecule here, a VDC, a VAR2 drug conjugate. We also made second generation variants of this, where we took a clinical tested payload, the MMAE, monomethyl auristatin E, that is the same payload that is on EV that you use in bladder cancer now. And this VDC here also could eliminate, this is prostate cancer, all prostate cancer cell lines, and you could completely eliminate tumors in mice with this VDC with no apparent toxicity. So that was all great. But the half-life of this protein was not great. So the half-life in serum was about 10-12 minutes. And we did all the tricks in the book to extend that half-life. We pegylated, we coupled albumin to it, a lot of different things. We could extend it to maybe 30 minutes, 45 minutes half-life in serum. And then there was the problem of stability. The stability of the protein batch was not great. And the manufacturing was problematic because there were batch-to-batch variations that we could not completely control and that we still today don't understand. So for that reasons, pharma did not want to take it on, take it further into the clinic. They didn't want to risk a new therapeutic like this on top of a new target. And the risk of a malaria protein fragment into a human, although our data suggested it was not immunogenic. It was not enough. So we had to change our strategy. So instead we used the malaria protein to purify raw amounts of oncofetal chondroitin sulfate from primary tumors and placentas. And then we ran 4 of the largest phage displays in the world. 1 in the US, 1 in China, 2 from Europe. And we went through 10 to the 10th phages, searching for binders that could bind oncofetal chondroitin sulfate without the protein attached to it. So only the glycan. And we got 19 clones out of 10 to the 10th phages. And after down selection of those 19 clones, we ended up actually with 2 clones that passed the negative selection, could not be competed with other glycosaminoglycans, were able to be competed by the VAR2 protein or purified chondroitin sulfate. So 2 clones, and those 2 clones are now in all these different types of antibody formats that you can see here. Okay, so the antibodies are highly tumor specific, just as the malaria protein. This is binding to different types of primary human tumors, and they bind very nicely to any kind of human tumors, both primary and metastatic lesions. This is an example for prostate cancer, where on top we have a generic, you can say generic non-specific chondroitin sulfate antibody. This binds all chondroitin sulfate. Down here we have one of the cancer-specific chondroitin sulfate antibodies. It binds very early lesions in pancreatic cancer also. This is an animal model of pancreatic cancer called the KPC genetic model. It binds colon, breast, lung, prostate, bladder. I have no bladder on here, but bladder is part of it as well. And then it also binds circulating tumor cells that floats around in serum or in plasma. And it's a truly oncofetal modification. This is embryos from mice. And the embryos there are positive, as well as the invasive fronts of the placenta. And when the mice then get old, then they lose the modification. It's not expressed anywhere in the mouse, just as we see in humans. And it's not only mice, it's also canine cancer, osteosarcoma, highly positive for this. And these antibodies, they are able to directly seek out the tumor in animals. This here is I think it's a pancreatic cancer model, this one here. This one is a neuroendocrine prostate cancer model. And it goes directly to the tumor, and it's not present anywhere else in the mouse. It penetrates the tumor very, very deep. This is tumors taking out of the neuroendocrine prostate cancer model. And then, because we have a small tag on the antibody, then we can detect it in tumors using immunohistochemistry. We can see where the antibody is. So over here, all this is the antibody we are staining for in the tumor. So now we have these different formats that we are working with. We have the ADC format. We can also put radionucleotides on it. I'm going to show you a little bit of that. Then we have immunocytokines, bispecific immunocytokines, and then we have bispecific immune engagers, so BiTEs, that are able to recruit T cells in one end and bind the Oncofetal Glycosaminoglycans. And half a year ago, I think we published the first CAR T paper with this, where we have CAR T cells also that can recognize Oncofetal Glycosaminoglycans. Just a few examples with the antibody here, or the ADC. So the ADC we make is a monovalent IgG format. And the reason for that is that it's smaller than an IgG. So it penetrates much deeper into the tumor than a regular IgG. And then we have engineered the antibody so that we can put exactly 2 toxins on it. So all the antibodies will have a DAR, or a loading of 2 toxins per antibody. And these ADCs, they completely eradicate any type of tumor that are positive for oncofetal CS in 1, sometimes 2 injections of the antibody, with no toxicity in rats and no toxicity that we can see in hematopoietic cells. The drop in neutrophils here is related to the payload and not really to the antibody titers. Something you see with all ADCs with MME. Just another example. This is lung cancer. And it doesn't matter what kind of payload you put on. You can put MME on, or you can put DXD on. It doesn't matter for this. All the payloads work very nicely. This is an example of the bispecific immune cell engager, the BiTE. This is a T cell engager. And this is an immune competent syngeneic model of breast cancer, the 4T1 model. So here, the T cell engager can also completely eradicate tumors very nicely. And if you try to implant a tumor after this, then it will not take in the mouth, because the mouse has developed memory against the tumor cells after the first hit. So this is just a few slides on the BiTE that moves forward, just to show that it binds tumor cells up here very nicely, and it also binds CD3 cells. And this is the kind of serum half-life we would like to see in a therapeutic candidate. Forward. So, about a year ago, we decided to take a step, what you could say, an intermediate step between phase 1 to clinical trials or the preclinical work that has gone before. And that is a phase 0 microdosing imaging trial we basically just jack in one single dose of the antibody linked with a zirconium isotope that we can pick up in PET scans. And this trial here is going on in Holland, and it's all comers, all solid tumors coming. And we just got the interim report actually from this trial here. This is a trial run by the development company, R2 Pharmaceuticals. And we have very, very clean PET images here that detects all tumors that we have had in so far. Most of them have been non-small cell lung cancer, but we also had a rectal cancer patient up here. And the interesting thing was that we actually detected a prostate lesion in that patient that they didn't know about. That was later confirmed by a biopsy. So that was kind of very gratifying. So at the moment, the C9 antibody ADC, so with the MMAE, is currently in GMP production. And I'm hoping that we will have that ready in the last bit of this year so that we can start phase 1, 2A clinical trials in first quarter in 27. Okay, so this is the first summary. I have shown you that malaria tropism can teach us something about glycosaminoglycans in cancer. And also that many types of cancers, they express this oncofetal glycosaminoglycans all over tumors, also in the stroma. And we have used this malaria protein to purify source materials so we can create therapeutic antibodies. And these therapeutic antibodies, they work very nicely. We can make different formulations of therapeutics, and in PET-CT trials, they go directly to the tumor with minimal binding in normal tissue. Okay, so now I want to just take a step away from chondroitin sulfate for a bit and talk about ADCs. Because in my mind, we don't really know how ADCs actually work yet. Although it's kind of the elephant in the room, because when you go to conferences, ACR and all that, there are ADCs all over the place. And they all tell the same kind of story on how ADCs are supposed to work. And they're supposed to work like this. They're supposed to bind a tumor target on the surface of the tumor cell that captures the antibody drug conjugate, triggers an internalization of that antibody into the acidic compartments of the cells. So that's the late endosomes, lysosomal compartments. In there are acidic hydrolases, catepsins, enzymes, proteases that are able to cleave the linker between the antibody and the payload. And in that way, you release the payload that can then go to the nucleus and do its job. That's the story. That's how ADCs work. If you read any textbook, this is how it's described. Okay. Keep that in mind. There are, so this list here is already outdated. I think there are 16 ADCs approved by FDA at the moment, with different payloads, targeting mainly these targets here. So there are few targets that are exploited by ADCs, but different payloads. Okay. Some payloads are microtubule disrupting agents, so like MMAE. There are also topoisomerase 1 inhibitors, like DXD, for example, that I also showed you a mouse model with. And then there are also some alkyliding agents like PBDs, but they tend to be quite toxic. Okay, So here is the first mystery point with ADCs. There are no difference in patients whether in terms of maximum tolerable dose or recommended phase 2 dose, whether you use an ADC with a toxin on it or you use just the toxin alone. Everyone knows it, but nobody really talks about it because it doesn't fit that dogma I just showed you. That you have to be internalized, and it's very specific. There's no difference in humans whether you use an ADC in terms of maximum tolerable dose. Keep that in mind. There is a clinical benefit of ADCs in any type of cancer they are deployed compared to if you use the drug alone. So you can dose exactly the same. The ADC has a clinical benefit. Okay? Less than 1% of ADCs actually reaches the tumor in a human body. So this is a study where they have taken some of the most popular ADC antibodies used and put a zirconium or a radiolabel on, done PET scans, quantified. So you have 0.1% to 0.9% of the ADCs, they actually reaches the tumor in the body. The ADCs also work in ultra low target expression. The trastuzumab DXd has just been approved in ultra low HER2 tumors. And what is the definition of ultra low? It is that you have barely noticeable IHC staining on tumors in less than 10% of the cells. So it means basically negative. But the ADCs, they also work there. So that to me is a mystery. And it tells me that something about ADCs that we don't quite understand yet. And this kind of mystery here has also been driving the notion, or at least the hypothesis by some people in the field, that ADCs are just sexy chemo. So imagine that the difference being that you are able to load chemotherapy into patients and retain it in circulation with a slow release mechanism. So maybe that's what's going on. But the truth is nobody knows actually. This is highly possible. So I'm looking forward to one of you at some point doing the GFP ADC trial. Taking, for example, a GFP antibody, put on toxic payload, and use it head-to-head against something else. See what happens. Pharma will not do this experiment. It will have to be in academia. But that's something we don't understand. I hope you appreciate that. Okay, so this is basically just what I've summarized here, and what I see as a controversiality in ADCs. Okay, so I don't have to show this prostate cancer stuff with you, but I'm going to give you an example of how oncofetal CS is regulated in tumors, and we're going to use prostate cancer as an example. At the Prostate Center, Y.C. Wang has developed this phenomenal model of prostate cancer, where he takes a treatment-naive PDX tumor, put it into a mouse, castrate the mouse, and then that PDX tumor will relapse, just as they do in humans, and then they will slowly progress into neuroendocrine prostate cancer. That's the endpoint. And we can take samples out on the way, sequence, characterize. And what we did was that we transcriptional sequenced these tumors and looked for the enzymatic machinery that drives chondroitin sulfate synthesis. And that is a complex system. There are more than 20 enzymes involved, and they are redundant. That's why they are very difficult normally to interrogate. So we were very lucky here, I have to admit, that one of the top hits were CSST11 came out. This is a sulfotransferase that sulfate, chondroitin sulfate at a very specific site that is important for oncofetal chondroitin sulfate. Sits up here, the 4-sulfation. And we could see that CSST11 was completely inversely correlated with AR activity, both in experimental models, but also in patient cohorts. This is the University of Washington cohort, complete inverse relationship. And we could also see in our own patient cohorts that patients that have been on ADT, they come back with high levels of CSST11 expression. That is because AR binds a repressive element in the first intron of the CSST11 gene. This is something we could see with chip sequencing, and we could map that specific binding site in CSST11. So when you lose AR signaling, then you hyperactivate transcription of CSST11 that then drives the modification that is characterizing oncofetal CS. And what we could do here is that we could, put them in androgen deprivation, then CSST11 comes up very rapidly. Then we could give them the hormone back, and then it drops down again. And then we could, on top of that, give them enzalutamide. Then you counter-rescue the system and brings it up again. This is really the proof that this is a purely AR regulated. And this leads to increased levels of oncofetal CS. So we can see that in a glycomass spectrometry, where we can specifically detect this 4-sulfation that is put on by this enzyme CSST11. And when we look in these samples here, both in clinical samples, but also in samples from the mouse model, but also in cell systems, then we can see this relationship that CSST11 is associated with high levels of oncofetal CS. That also translates into differential binding with the VAR2 protein, the malaria protein, that we can still use as a reagent, of course, to characterize and detect oncofetal CS, although we cannot use this as a therapeutic. That shows the same. We also have ways to visualize the glycocalyx, so the sugar coat around prostate cancer cells. And we can do that by incorporating an artificial sugar ring into chondroitin sulfate that we can click a green fluorescent label onto. So it basically means that we can get a snapshot photo of how the chondroitin sulfate glycocalyx looks like when we manipulate, for example, AR or chondroitin sulfate directly. In prostate cancer, we cannot knock out that enzyme. And believe me, we have tried many times. So instead of knocking it out, we did a recombinant cell line that expressed an enzyme that degrades chondroitin sulfate after the fact. And in that way, we can deplete chondroitin sulfate in prostate cancer down to, I would say, around 20% of normal. And that is just enough that they survive and behave fairly normal. But it is enough so that we can ask research questions on function of chondroitin sulfate. So these cell lines here, they are unable to move and have motility after androgen deprivation. That was the first very strong phenotype that we observed. And when we took mice, we took these LNCAP cells, one that did not have chondroitin sulfate or reduced chondroitin sulfate, and one that had normal levels of chondroitin sulfate. Then we established tumors, and then we castrated the mice, waited for them to relapse. So this is before castration. They grow pretty much similar. But after castration, only the wild type tumor with normal levels, high levels of chondroitin sulfate, were able to relapse after castration. So that really told us that chondroitin sulfate is very important for prostate cancer in the context of AR inhibition. This was the proof we were looking for. And we can see that very nicely in all these advanced prostate cancer TMAs. This is the NHT TMA that we have. They are basically all highly positive for oncofetal chondroitin sulfate. This is the neuroendocrine prostate cancer TMA, also very, very high. So this really confirms what the biology shows us on mechanism. Okay. So the last summary here is that prostate cancer, they increase oncofetal CS during progression. It's AR regulated, but it's a negative regulation mechanism by AR. So when you put them under androgen deprivation therapy, or if the tumors lose AR for whatever reasons, then you will get an even higher expression of oncofetal CS. So that, of course, is a very strong rationale for including prostate cancer in the upcoming phase 1/2A settings. So that's what we are hoping to do in this context here. Okay, so lots of people over the years have been involved in this. Put some of the key people in bold that has been really key drivers of the oncofetal chondroitin sulfate projects over the years, working on ADCs, but also on CAR-T cell therapy and bispecifics, so on, so forth. And of course, lots of collaborators in the clinic here and BC Cancer and companies. So over the years, this has been very expensive. This is just the oncofetal chondroitin sulfate projects over the years. And most recently, we this year got 2 big grants, one CRHR grant, about $1 million, to specifically look at qualifying the ADC in advanced prostate cancer. So a grant specifically for that. So there we will try to build the preclinical package that we need for that. Then we also got the Terry Fox Foundation grant with Peter Morgan. Project 3 in that Terry Fox grant is basically the same in bladder cancer, but there we will also look at the BiTE, so the bispecific T-cell immune engagement, and see how that performs in bladder. With that, I will stop here and happy to answer any questions you might have. Thank you very much.